Welcome, my digital pathology trailblazers. Today, I would like to take you to the Journal of Pathology Informatics to discuss a paper, Development and Technical Validation of Artificial Intelligence Model for Quantitative Analysis of Histopathologic Features of Eosinophilic Esophagitis. This paper is authored by Louisa, Lindsay, Hannah Kaisa, Thomas, Ville, Donaka, Maria Camila, Erin, Yao Hong, Priyadarshini, Ameya, Wanani, Imad, Kartik, Taufik, Rish, Catherine, Christopher, Rondell, and Roger. And those authors are from Mayo Clinic and from Aiforia Technologies. The model was developed in the image analysis platform Aiforia, and it was a supervised deep learning model with annotations. But what is cool about those papers that show you development of those models, the cool thing about them is that the pathologists have to translate a pathology scoring into image analysis. And this is the coolest thing about those papers. Why is it even done? We have the pathology scoring and then the group wants to develop image analysis. Well, they want to have it quantitative, they want to have it reproducible, and they want to do some statistical standardized analysis. And that's why they develop an AI model for digital pathology to reproduce it with an image analysis software and to quantify the components and to actually compare different things in this model. And here in the methods, we already start with an interesting thing. They tell you exactly how many objects they annotated and what area they annotated. This is really cool because there's always this question in those model developments, how much do I need to annotate? So over 10,700 objects, that's a lot of objects. Whether 56 millimeters square is a lot, I don't really know. But if you are doing this, you can compare how many millimeter squares you have with this very paper and you know, okay, they annotated more or less. The software they used is our Aiforia, the cloud-based deep learning artificial intelligence platform. And we had a lot of those objects, but it was done on 40 carefully selected biopsy slides. So not so many slides. So you had to annotate a lot, but you didn't need such a huge amount of slides, 40 slides, and they were already able to develop a decent model. And we see here that the subset of cases was reserved in an independent test set. A couple of years ago, this was not standard that you had a test set. The life sciences community didn't know that you need a test set to actually check those models, and they were reporting data on the training set. So we now know we need a test set. The abstract doesn't describe the exact features of the esophagitis, but we're going to dive into it in a second. And not only did we have the test set, there were also 5 specialized gastrointestinal pathologists who were scoring those images blindly, independently of each other and of the AI. And the performance of the AI model was similar or non-inferior to the GI pathologist. It was a pretty high F1 score, 94.5, 94.8 for AI versus human, and from 92.6 to 96, human versus human. Another interesting thing is they rated accuracy in a very pathology typical way. So they use a scale from 1 to 4. So 1 was perfect or nearly perfect, and it was 95 to 100, no significant errors. Whereas 4 was insufficient, but it was just under 70%. So like in the normal world, you would consider something that's 70% a pretty high score. Here, this is insufficient. So that's also an interesting reference when looking at accuracy. Where do I draw the line? So they drew the line at 0.7. Here we get into the classes. They detected tissue and the rating was very good. And spongiosis, also very good, 1.15. Basal layer, surface layer, lamina propria, collagen. So all of them were in the very good or perfect, nearly perfect range. They also detected degranulation. This was a little worse, 2.23, good or very good, but that is still okay. So in the conclusion, the thing worth noting is that the tool showed great performance in recognizing various histologic features. So all those features were classes in the model, and the model was trained to recognize those classes, and it recognized it as well or better than a pathologist based on the pathologist's annotations. And this model is a proof of concept that you can do it with image analysis, and they call it an important step in creating an accurate and reproducible method. I love the way they describe how the AI model was trained, because it super nicely describes how Aiforia platform works and how it's actually pretty useful to design those supervised model for pathology. So here the AI model was trained as a set of nested individual convolutional neural networks called layers. This is how it's called in Aiforia. The individual CNNs were trained independently of each other, but were executed in a progressive fashion so that the topmost parental CNN first analyzed the slide, and only the areas identified were subsequently analyzed by the following CNNs, first child, and so on. Each class was designed to identify single or multiple classifiers called classes, and annotations were provided as training. So how it works in Aiforia, and we're going to see it on the images later. They have beautiful images here, and we're going to go through all of them. But you detect the tissue first, and then you detect something in the tissue, and then you detect something in the tissue, and then you have a layer tree of different layers. So this is a very nice description. CNNs nested in each other, trained independently, but only deployed in the result of the previous CNN. So let's look at the classes. We have tissue. Tissue is always the first thing. It's so funny for a pathologist to have to detect tissue, but in the whole slide image, there are plenty of white pixels as well, which constitute this image. So you have to have the tissue selected from the background. So this is always the first layer. And what's super important is to define what your tissue is. So it's not everything, right? Their tissue was total esophageal mucosal tissue, excluded columnar and non-squamous mucosa. So these parts, the columnar and non-squamous, were part of their background. So they didn't even bother detecting it. And we have the outputs here. And whenever we're working with regions, we will have millimeter squares. Here we are talking about semantic segmentation. If we have both area and the count, we are talking about instance segmentation. And yeah, I do see here only count. This is object detection. So going back to our layers, we have tissue, we have intracellular edema, granules, surface layer, basal layer, lamina propria, collagen, eosinophils. This is eosinophilic esophagitis, so we do need to detect eosinophils, lymphocytes, and squamous and nuclei. So these are the classes, and here are the layers. So this first layer only has one class. The second layer has two classes. Third layer has 1, 2, 3 classes. Fourth layer is just one class, and 5, we have 3 classes. I wish I could see the whole layer tree in the image. And we have the morphological features described. So this table is the translation of a pathology into computer vision. So basically those morphological features, we assign them a class, and this is into a layer. So this is your translation here, and then we're going to see it on the images. So image A is the original image without any markups, and here we are starting to see markups. So the green guys are eosinophils, and we see them here and here. So this is our native image without any markups. This is H&E, and these red guys are eosinophils, and we can see them here on this image as green dots. So all those green guys are automatically detected eosinophils. In image C, we have these are eosinophilic abscesses. And on image F, we have their automated detection, all those green guys. And what you can also do once you have those objects detected, you can do spatial analysis. You can measure distances. And this is what's happening in those yellow circles. There is a spatial analysis component to it. So the red guys this is not image analysis. This is native image. And by native image, I mean nothing with a markup. There is no image analysis markups. The green dots are showing us with image analysis those eosinophils. Same on image F. And here we also have the blue guys. Blue guys are lymphocytes, another type of immune cells that is a component of this esophagitis. And the red guys are just nuclei of the squamous epithelial cells. So all these guys who don't belong to any immune cell population are also detected, so that the model can learn 3 different type of nuclei and consciously, quote unquote, distinguishes between the epithelial nuclei and the eosinophilic and lymphocytic nuclei. Image A is the native image of esophagus, and here we have different layers. Different colors are segmenting. Different colors mean different parts of the esophagus. And then once you have those different parts detected, you can check if you have those cells that you were training before in those particular regions. So what are those regions here? So the regions here are in blue is the mature superficial epithelium. So here you can see the light pink color, and the more purple color that corresponds to the green layer here is the basal layer, the younger part of this epithelium. What's shown in yellow here are dilated intercellular spaces, and red is lamina propria. This is connective tissue. These are images of biopsies with image analysis markups. And the first one is density heat map of epithelial nuclei. And you can see that here in red is the highest density of nuclei, which corresponds to our basal layer. Makes total sense because in the basal layer, like you saw on the native image here, there's more of those nuclei, so the density is going to be higher. So we see it here in the heat map, and it corresponds to the appropriate class. And here on the top we have normal squamous mucosa, so this would be the ratio of the basal layer to the superficial layer, whereas here at the bottom we see that there is more green than there should be. This is classified as basal layer hyperplasia. And even if this model would just show us that, it's visually so much easier to distinguish than in the native image, right? This, like, it blends into each other, whereas here you can distinguish very nicely blue and green. And actually you can pick whichever colors you want in this software. The yellow class here is called spongiosis. This is also part of the scoring. This must have been a tricky one to train, but it's doing a really good job. And you can immediately visually see A has less than B and B has less than C, meaning when we're going to be grading them, this is going to have lower severity grade than the B one and the C one. So here this was mild, moderate, severe, but it's so easy to see if you have this yellow markup. Like, you don't need anything else. You just look at it and you can immediately grade. Here we have the assessment of lamina propria and collagen. I like very much how they solved it. They have 2 classes. They have lamina propria, which is this thing, all this thing, and this as well, that is underneath the epithelial cells, but they also have collagen separately. And you can see a lot more green stuff here on the right than here on the left, right? If we only would talk about lamina propria, this is bigger area than here. But then what they did a ratio of lamina propria to collagen area, and they called it fibrous tissue density score. Really sophisticated model. So you can appreciate it on those images. This is the native image. Here we have segmentation of lamina propria. C is the collagen. And here D, we have the superimposed collagen and lamina propria, and we see how much collagen there is within lamina propria. Even visually, if you don't calculate, you see it's a lot. So the area of one high power field in one microscope does not necessarily equal the area of another high power field from another microscope, even though we're looking through the same objective. But here, the high power field. So in this publication, the area is 0.24 millimeters square, because those eosinophils need to be counted there, right? So how do we solve that with tissue image analysis? We can do the tiling. We can use tessellation. What's tessellation? Tessellation is tiling, dividing it into tiles, and those tiles would be approximately 24 millimeters. But the problem here is that you can see that this tile, for example, is mostly non-tissue. So this is not really that helpful, but there is a different method called Voronoi a method that helps you divide this into fragments that have more or less the same area. This method is used in biology, and the authors here adapted it to pathology as well. So each of these guys here have more or less the same area of 24 millimeters, and they do not overlap and they do not have gaps. And then what you can do, you can quantify whatever your metric is. So here the number of eosinophils in each of those fragments, right? And then you give it a grade, 0 to 3. And that's how you quantify your high power fields on a whole slide. So that's pretty neat. There's a lot of translation necessary to take the pathology scorings done with a microscope and translate it into a computer vision approach. And could we not do a different computer vision approach? Yes, we could. And here different features are added. But if we want to compare it to something that we know is working or is standard of care, established method, we have to compare it to this visual scoring. That's why this computer vision approach is basically mimicking what a pathologist is doing under the microscope. But it's doing it in a more quantitative way. It's doing it on a whole slide. So we already have a lot of added value, even though we're mimicking the visual approach. Are we going to ever get away from the visual approach? Probably, but it's going to be a stepwise approach. Maybe there is going to be a way to streamline this later, but for now, very nice proof of concept. So what was the performance of the model compared to annotations? That's always pretty interesting to me. And the metric I usually look at is the F1 score. This is the most comprehensive metric. And in most of the cases here, it's above 90%. So let's check the ones that are not above 90%. So this guy here, well, who's that? This guy here, these are the granules. Okay, so granules were not that well, and dilated intracellular space were also not that fantastic. But other than that, everything above 90, not bad. Here also, AI versus human, human versus human for all the cell types, for eosinophils, lymphocytes, and squamous nuclei. And again, F1 score, everything above 90%. So yeah, good model to conclude. This is the first model for quantifying eosinophilic esophagitis, mucosal eosinophilia related histologic findings. And they recognize that they have to do a little bit better with the eosinophil granules and the comprehensive evaluation of collagen patterns. So there is room for improvement, like with all of those models, but there's already value. So fantastic. You stayed till the end of me discussing this paper. This means a lot to me. And that means you're a real digital pathology trailblazer. So I have something I think you might like. It's my Digital Pathology Starter Kit. It's a bundle of curated resources about digital pathology. It's like a mini course that covers more aspects of digital pathology, not just image analysis. So go ahead and grab your copy in the link below, and I see you on the other side.